Hello and welcome to the journal webinar series of the American Society of Agronomy, Crop Science Society of America, and the Soil Science Society of America. This series features the latest research published in our society journals. I'm Emily Miller. I'm the journal manager for the Plant Phenome Journal, and I want to thank you for joining us today at the webinar, Pedigree-Management-Flight Interaction for Temporal Data Analysis. Before we begin, I would like to note that questions can be entered in the question box, and the moderator will address them at the end of the presentation. This webinar will be recorded, and the recording will be emailed to all attendees after the session. With that, let's get started. I am happy to introduce our moderator for today's webinar, Dr. Seth Murray. Dr. Murray is currently a professor in the Department of Soil and Crop Sciences at Texas A&M University. Dr. Murray's research interests focus on improving the productivity, sustainability, and quality of agricultural production through scientific research and development, mostly in maize. He founded and served as editor of the Plant Phenome Journal, and he has served in several society leadership roles, including currently serving as the president of the Crop Science Society of America. All right, I'll hand things over to Seth.
Thank you, Emily. Well, today it's my pleasure to introduce currently a postdoc with me, formerly a PhD student, Dr. Alper Adak. Alper is a leader in phenomics and data analysis. He's really propelled at least my program forward, and I think the whole community forward with some of his innovative discoveries and methods and procedures. He's a great mentor of a lot of new graduate students in this area, and he has won the Vice Chancellor's Excellence Award for graduate students and the North American Plant Phenotyping Network Graduate Award last year, and he'll be presenting at the NAPPN meeting this year. He's a native of Turkey. He got both his bachelor's and his master's at Akdeniz University in Turkey. And so I guess without further ado, Dr. Adak, if you'd like to present.
So yeah, thank you very much for this nice introduction, Dr. Seth Murray. And I also would like to thank dear Emily and the Plant Phenome Journal for this amazing opportunity. And I am sharing my screen right now. And yeah, I believe my presenter screen is right now. Sure. So we recently had a chance to publish an article in the Plant Phenome Journal called Pedigree-Management-Flight Interaction for Temporal Phenotype Analysis, as well as Temporal Phenomic Prediction. And in this manuscript, myself is the first author, and the second author is Stephen Anderson. He also finished his PhD in our lab, and right now he's working in the industry, and he helped a lot in developing our mixed model. I will be presenting in the following slides, and Dr. Seth Murray, he is a full professor at Texas A&M University, the Department of Soil and Crop Sciences, and he is the corresponding author in this research. Before moving forward, I just would like to briefly talk about our fundamental steps in data processing, in field-based high-throughput phenotyping processing. That includes different drones, sensors, image processing, and data extraction, and many other software we've been using since almost 2017. If I start with here, so this is just basically drone flight operation, and we are flying drones over our breeding population, and these are just the small JPEG or the TIFF file belonging to a small part of our experimental sites, and we are using the ground control point at the beginning of our first try, because the drone we were using didn't have the RTK option. RTK in this case, briefly, if there is a RTK option in the drone, we don't need to use the ground control point, but if there is no RTK option in the drone, we have to use. I am telling this thing because right now we have bunch of collaboration from different universities, and they are sharing their images with us. And unfortunately, the main problem we were facing with is lack of or the absence of the ground control point. And if there is no ground control point, unfortunately, we are having issues in making orthomosaics and 3D point cloud data that cause erratic results at the end. So this is why I would like to emphasize the importance of the usage of the ground control points. And then once we merge all of these raw images, we are getting 2 different files at the end. So we are using some software such as Pixel 4D or Metashape, and then to merge all of these raw file, and we are getting the orthomosaic. So this is the one of example of the 3D point cloud data, and we are using it to extract the plant height data. And this is just the top view, and this is the lateral view. And then the following process is to process the data. And this is a very well-known procedure, but in order to briefly explain what these are, is that so DSM is plant plus ground, DTM is ground, and we are getting the CHM by just doing a simple extraction, the DTM from DSM. And then we are creating a shape file. And then we are merging the shape file with the 3D point cloud data, and we are cutting each row plot as it's seen here, and we are calculating the plant height. So as for the extracting the vegetation index by using the reflected bands information such as red, green, and blue, we are using the TIFF file as it's seen in here. So this is a little bit different than the 3D point cloud data, but it contains the same information in it. But we in general, we prefer to use the orthomosaics for vegetation index. That doesn't mean that we cannot use this thing, the orthomosaics, for plant height extraction, but this is just, this became our routine through the years. So this is just a simple orthomosaics, and this is our range row-based experimental design. If you zoom in, you can see the 2 row plot. In general, we are growing our plants 2 row plots, so we can easily see that, okay, maybe these 2 row plots are just one genotype, for instance. And these are the row plots, and this is the range. And this is a typical TIFF file, and then we are removing the soil. As you see, there is no soil in this picture. And then we are using the field imager, by the way, in R. And then again, we are using the same shape file, and we are merging this shape file with the orthomosaics, and this is what it's seeing. So when we zoom in the picture, we are seeing that the shape file are covering each row plot like that. And then we are getting the vegetation index per each row plot, and then we are saving this in the CSV file at the end. And this is another picture showing the another perspective of our process, field-based high-throughput phenotyping process. So we are using mainly multispectral and RGB camera with the drone, and we are flying our drones over our populations, and we are capturing different growth stage of maize growth. So we are interested in maize, but we can capture any growth stage of any crop by doing the same application. So briefly, what I can show in here is that we are capturing the first stage of growth, which is establishment stage, vegetation stage, flowering, grain filling period, and the senescence. We can easily see all this growth stage and the temporal change in this growth stage. So when we move to C part, so this is the another view of the shape file and the orthomosaics. When we merge them, and if you look at the top view, this is what we can see. So these are just a zoom in picture for the same area across different time point of flights. So when we look at these row plots, we are not seeing any differences based on the naked eye. And when we look at this picture, what we are seeing is, again, there's some growth pattern, but there is no significant differences, again, based on naked eye. But when we follow through the time, we are seeing that, okay, this genotype is senescent earlier than the other. So, and then different flight time contains the different variation of the same genotype. This is why temporal variation might be important. And the D part, I will explain, I will delve into this analysis in the further slides, but briefly, we are using the 3-way interaction, genotype, flight environment, and their 2-way and 3-way interaction. This is for plant height example. So on the x-axis, we are seeing the flight date as days after planting unit, and y-axis are the plant height, and these are the plant height change across time point. By the way, I would like to mention the unique collaboration of the Dr. Jacob Washburn from USDA, and he shared this data with us. And then this is the temporal plant height example. This is the NGRDI. NGRDI is a simple vegetation index. If I'm not wrong, the calculation of NGRDI is simply green minus red divided by green plus red. So, and then this is the temporal change in NGRDI through the time point. And if you look at the E, this is out of scope right today, but once we have the genomic information of the data, genomic information of the population of interest, and then if we have the temporal phenotype data for the same population, we can merge these two information to better understand the loci or candidate genes that are playing a major role on plant growth. And F is the temporal phenomic prediction. I will be mentioning that at the end briefly. And this is another view of the 3D point cloud data. If we look at this part, this row plot looks taller than the other one, and this is one of the example of 3D point cloud data. So right now, I would like to introduce our simple, but we believe the efficient mixed model to analyze the temporal phenotype data. So in this research, we use this data in the published article in the Plant Phenome Journal, and we used two different experiments. In the first experiment was grown in 2017, and then we used a 220 maize hybrid grown in both optimal and the late planting. And we flown the drone at 48, 63, up to 112 days after planting. So in 2019 experiment, we used 100 maize hybrid, and again, they were also grown in optimal and the late plantings, and we were flown. The flights were flown at 39 to 96 days after planting. We wanted to propose this mixed model since we have seen that the many researchers tend to analyze the single time point differently by disregarding the flight time, but we have seen that the major variation is happening through the flight time. This is why we wanted to advertise and publish this simple but efficient mixed model. So as we see in here, this Y is any temporal data. It can be plant height, which is CHM in here, and the NGRDI. We use just two examples, but it can be any temporal data coming from the drone flights. And new is the grad mean, pedigree, flight, and management. Pedigree can be any type of population you're interested in, and the flights is the flights, but we are using the flights as days after planting, but still categorical. And then the management can be environment or different management, but in this case our management is optimal and the late planting, but the management can be environment A or B, and they are two-way and a three-way interaction. So when we run the model for temporal plant height in 2017 and 2019, we are seeing that the green explains the highest percent of variation, which is flight. So we're not surprised for that. And when we look at the NGRDI, one of the vegetation index in this case, we are seeing pretty much the same scenario. Flight or any component containing the flights in it tend to explain the highest proportion of total variation. So this is why we think that using the three-way, including the flight as a novel component in the mixed model may help to understand the plant growth by using the temporal phenotype data. So followingly, what I can advertise is the temporal repeatability or heritability. So this is very, again, simple heritability calculation, but the only difference is we are including the flight in it. So A and B in this case is the number of management and the number of flight. So and then by using this simple ratio, we can also calculate the temporal repeatability for the temporal phenotype data, such as CHM and NGRDI here. This slide is showing the results of pedigree flight management component. So if we look at the A, what we are seeing is that the LP and OI, by the way, late planting and optimal planting in 2017, we are seeing that the same population produced the faster and the greater plant height during the vegetation stage as compared to those in optimal planting. So this is pretty much accepted because optimal planting in Texas for maize is around first week of March, and the late planting is 1 month later than first week of March. So this because of the day length situation, greater day length and the greater growing degree days and many other things, we are seeing this scenario, which is the late planting had produced the taller, faster and the greater plant height as compared to those in optimal planting. We have seen pretty much the same scenario in 2019 as well. So, but what I would like to highlight in here is that in order to understand the plant growth and the change of the plasticity in the temporal phenotype data, we can use the results of pedigree flight management interaction in our mixed model. So in the B and D section, what we are seeing is that in this case, we can also visualize the results of flight management interaction as well. So I put the yield information of the late planting and optimal planting for 2017 and 19 hybrids. What we are seeing is that late planting had a lower yield than those in the optimal planting. And this is the whatever variation happening in plant height is a kind of like a early indicator of the yield, what we are going to see at the end of the season. So basically, the temporal plant height change, like what we are seeing in the late planting, is kind of like an undesired temporal trajectory, for getting the low yield. And this was for plant height, and this is for the NGRDI example. Again, when we visualize the pedigree flight management interaction for NGRDI, we are seeing the different temporal trajectories for the different management. Again, LP and OI is the late planting and optimal planting respectively, and we are seeing the change in temporal NGRDI score for 2017 hybrid in A and 2019 hybrid so the temporal change in 2019. So again, we are seeing that there is some change happening before or after the flowering time. By the way, this dashed line is showing the flowering time, and also this is another temporal trajectory we can visualize by using the pedigree flight management interaction. So this example is not included in the R manuscript, but I would love to show a few more examples about how to use the R simple mix model. So again, we use the same mix model, genotype, flight, environment, and their two-way three-way interaction. So in this example, we used another different experiment, and then in this one, 220 maize hybrids were grown in optimal in the drought experiment in 2020. These are the genome field projects hybrid. And then we flown the drones many times, as you can see on the x-axis, and the VARI, this is another vegetation index on the y-axis. And when we run the model for the same population, but grown in optimal and the drought experiment, what we are seeing is that at the end of the growth, there is a little bit variation because the VARI score decrease a little bit faster than those in optimal planting. So this is also another indicator that obviously we had the lower grain yield in drought trial. This temporal pattern is the indicator of the low yield. So basically, the results of genotype flight environment interaction can basically be an indicator of what type of yield we can get at the end. And this is showing the correlation between the VARI index and the yield. Yield on the x-axis are the same for each section. The title are showing the days after planting, 19 days after planting, and 2018 up to 117. On the y-axis, we are showing the VARI. So what we are seeing in here is that the correlation between VARI and the yield in optimal planting is changing between 0.56 to 0.553. The change is not that big. The reason why I'm showing this one is that the correlation between vegetation index and the yield can be also visualized by using the results of genotype light environment interaction component. And in here, what we are seeing is that correlation is not changing across time points. So if we use the temporal data of the VARI score belonging to every time point, is also can be important data for us to understand what type of grain yield, well, how much grain yield we are going to get at the end of season. So again, this is a totally different environment, the totally different data set and environment as well. In 2018, 515 recombinant inbred lines were grown in again optimal and the drought experiment. In this experiment, we have grown and we have flown the multispectral camera, and then what we are seeing is that the irrigated trail had a greater NDVI score as compared to those in drought experiment. So this is the phenomic prediction. I will be briefly talk about what it is. We are still developing this idea, but in this experiment, we used 415 hybrids grown in 2018 and 19. And this is our population structure. This is again a genome to field project hybrids. And in 2018, this is the population structure. I used the genomic information to show it. And in 2018, we have our yield was far higher than the 2019. When we look at the simple NGRDI score, we have seen big differences in the early growth, but not late growth. So whatever variation happened in the early growth may cause the differences between yields. So this is the reason why I would like to indicate the temporal, the change in the temporal variation in NGRDI happening in the vegetation growth and its effect on the yield. And this is my last slide. And in 2018 and 19, for the same population, were flown at the different time points. So what we have done in here is that we simply run the genomic prediction by using the genomic relationship matrix, and then we also run the same idea, but by using the phenomic data, by using the phenomic relationship matrix. So these are the CV1, CV2, CV0, and CV00. These are the potential prediction scenarios that are relevant to plant breeding program. But what I would like to highlight in here is that I use 3 different temporal time, which is from planting to V10 to 12 stage, from planting to flowering time, and from planting to anthosenescence. And we use the 36 different vegetation index and belong to these 3 different time points. So briefly, I would like to call your attention to CV1, which is the untested genotype in tested environment. That means that untested genotype means the genotype that was not included in the prediction model. So CV1 is typically refers to a prediction scenario happening in any public breeding program, for instance. So when we use the M1 is the genomic prediction, M2 is the phenomic prediction, and M3 is the combined prediction. We are using basically multi-kernel in M3. When we look at the, and then my x-axis in here is showing the different temporal time point. For instance, in this one, we use the 36 different vegetation index belonging to V10 to V12 stage. And then when we run the phenomic and genomic data, they perform pretty much same. And when we use the information of 36 vegetation index belonging to until flowering time, again, the genomic prediction and phenomic prediction perform same. But in these two scenarios, what we are seeing is that when we use the combined prediction, we are seeing a little bit greater prediction accuracy. But when we look at the CV00 prediction scenario, which is the hardest one, and indicates the untested genotype in untested environment, which is the genotype that was not used in the training population, and their prediction in totally different environment. And when we look at that, the phenomic prediction looks a little bit better than the genomic data, because genomic prediction accuracy is around 0.18, but when we look at the phenomic prediction, we are seeing slight increase. But I would like to highlight that in this section, we use the temporal data from planting to until we tend to be 12 stage. So we can predict the grain yield by using the earlier time point. But it does not really change when we use the all data, what we can see in here. Still, genomic prediction and phenomic prediction perform pretty similar, but when we use the temporal data belonging to vegetation stage in general, we are seeing that the phenomic prediction is working a little bit better than genomic prediction when it comes to predicting untested genotype in untested environment. So with this, I would like to finish my presentation, but my whole productivity and whatever research I conducted is coming from the team effort. So I would like to thank Dr. Seth Murray. He's our head and leader in here. And then I also would like to send my special thanks graduate student with us, Aaron DeSalvio, and then Mustafa Arif Arık. They are working a lot and they dedicate themselves, and they're producing a lot of phenomic data for us. And then also I would like to thank the other member like Colby Bass. He's taking care of pretty much everything, every planting in our lab, and the other previous staff members. So with that, I would like to conclude my presentation and happy to get any question if you have. Thank you.
Okay, well, thank you, Dr. Adak. We're having a little issue where I can't actually send chat to the attendees, so I'm asking Emily if she would be so kind as to do so, because I did respond to a couple of these with some journal links. But Alper, the first question that we received is, have we tried this approach for plant height in pastures? Would it be different due to the closed canopies in pastures compared to maize?
My first answer is that I really didn't try another crop than maize so far, so I don't have any robust answer for that. But again, I just need orthomosaics of different crops to examine the posture or the other things. But what I have heard from a professor from a Georgia University, I am very sorry since I can't remember his name, but what he said that biomass or biomass related traits, let's say in general, it can be predicted better than the grain yield, because EAV can capture biomass or biomass related trait better than grain yield, because whatever we are seeing is whatever we are using. So this answer might be a slight answer for this question, but again, I didn't try anything related to this question so far.
Okay, and I had typed out an answer, and I'll just read it briefly, but it's got a couple journal links. So a key for plant height is having bare ground or ground control points of known heights. So bare ground can be done before the plants grow, which is a best method if the drone has RTK with both XYZ accuracy of coordinates. Or bare patches could be included. So with maize, we have some bare ground that we can use to get those canopy height models. And so I put two studies in here. The other thing I mentioned is that Dr. Alex Thomason, who's now at Mississippi State, had some ground control points of various heights, so different, like a step, and he found that that could improve canopy accuracy quite a bit. So if you're going to do plant height in pastures, having ground control points of a couple different heights would probably help. So the next question was, how is the flight encoded in the model? Is it like 0, 1, 2, 3, or is it in another form?
Okay, so we are using days after planting unit. These are basically number. They looks number, but we are treating them as categorical variable in the mixed model. But this mixed model is just an idea of us. So there are other multiple options to analyze the temporal data. But again, this is just a simple mixed model, and we are treating the every flight data as a categorical variable in the model, like environment. So this is how I can answer this question.
Okay. Next question. I think the person may have been missing a word here, but our flight data can also be correlated to relative growth rate of plants. So can we, I think can we get the relative growth rate of plants from the flight data as part of the question?
Well, again, so I guess I don't know if another member in our lab did a similar stuff with what you have said in this question, but yeah, I mean, personally, I didn't do anything relevant to that, to be honest.
Yeah, and I'll just I put a link in here. Stephen Anderson published something previously in the Plant Phenome Journal where he developed a logistic model that captured the growth rate from the plant height. And based on that, you could get the relative growth rate by modeling the height over time. I mean, I think on Alper's data, he showed the progression of plant growth, and the relative growth rate, I think, would just be the amount minus the previous flight date, right? And you can even, once you model it, you can even interpolate between flights, and that will help figure out the growth rate. Let me see. Can you explain how you chose the vegetation indices? Why different indices for different data sets?
Yeah, so I'm getting that question very frequently. So I just look at the literature and see what the previous researcher used, what type of vegetation index were used previously. So this is my main source, to be honest, but we are consistent when it comes to using the same vegetation index across year. And yeah, so we are using pretty much 30 vegetation index, but of course we can downsample them. Only 4 or 5 of them are having a really good correlation with grain yield. But the important point I would like to highlight in here is that when it comes to RGB sensor, for instance, there are only 3 bands, and then you are limited to create the vegetation index by using three reflective bands, which are red, green, and blue. So well, maybe I can answer this question by giving a different example. So when it comes to genomic data, we are just filtering them based on minor allele frequency, and if they are not segregated, we are dropping them. So the same situation in the phenomic data. So the vegetation index are correlated between each other, but we are decreasing this correlation by using the temporal information of each of them. So this is how I can answer this question, I guess.
Okay, the next person asked 2 fairly complicated questions, so I'll ask them separately. First, does the multi-kernel model in the combined model account for the correlation between indices in different time points?
Your voice, I couldn't hear your voice.
Okay. So does the multi-kernel model in your combined model account for the correlation between indices in different time points?
We use the phenomic relationship kernel, like how we use the genomic relationship matrix or kernel in genomic data. So we are basically doing this is an issue, and I believe in both genomic and the phenomic data. But I guess the previous answer might be an answer for this question as well. So first, we are we are just using the different time points to see that to calculate the phenomic relationship kernel, but yeah, there is no any particular step that account for the correlation, high correlation, or yeah, the high correlation between vegetation index. But again, we believe that the temporal pattern of, or the temporal value of the vegetation index decrease the high correlation issue in the phenomic data. We believe that.
Yeah, and I'll just add, because there's been a common question about multi-collinearity. Alper produced a figure that he didn't show today that showed a genetic mapping population with, I think, 150,000 markers, and the multi-collinearity there was probably worse than what we saw with phenomic data. So the second question from the same person is, have you tried other models for combined genomic and phenomic prediction?
Last few years, we have spent lots of time for that. We collaborated with the statistician from, again, A&M, Texas A&M University. I didn't mention the detail of the phenomic prediction in here, but I can barely tell that. So we are using functional principal component where we are including the time point in it, and we are getting the functional principal component scores for each vegetation index that are segregating across genotype in environment A and in environment B. But we are not having the same situation for genotype. But we didn't try any other model, but we more like focusing on right now the application of the temporal phenomic prediction right now. Yes, so yeah, this is how I can summarize what we are doing right now. But first, we are using the FPCA scores, and then we are conducting the phenomic prediction by using the simple genomic prediction idea. We are using RK-HS kernel, for instance. So if that is answered for this question, I can tell that.
Next question. I have a general basic question. Why are we using two sensors separately, RGB and multispectral? Multispectral contains RGB, so why would we use RGB separately?
Oh, okay. This is there is no specific reason. We are trying. First of all, this is our main. It can be a short answer, but when it comes to detailed answer, the stitching. We are flying the RGB drones closer to the ground than the multispectral camera, because the stitching the 5 bands is taking more time compared to stitching the 3 bands. So based on my experience, what I can tell that regardless of what type of sensor you are using, the resolution might be more important than the sensor type we are using. So if I ask the question to myself, do I prefer to flying the RGB drones with the 20 meters, or do I prefer to flying multispectral sensors at 60 meters? So I prefer to RGB to fly at 25 meters, since it will provide them more resolution. So it depends on what you're looking for and then what research you want. So if you would like to calculate the NDVI, you have to fly the multispectral camera for sure. But if you're looking for them high resolution, you may choose the RGB to fly closer to ground. So again, it depends on what you are examining.
Yeah, and I'll just add that our group has been using a Phantom 4 RGB and a Phantom 4 multi-spec RTK, and so those two got flown on separate dates. So although I think we were using all the information per flight, but some days we only had an RGB flight, which, as Dr. Adak just explained, is higher resolution, and other days we just had a multispectral flight, which was lower resolution but included additional spectral information. So if you're thinking about phenomic data, both have a certain richness to them, but the multispectral has a little more richness because there's more bands. But that's why those were analyzed separately. The last question that I have in the list right now is, did you use repeated measure analysis in your mixed model for different dates of flight? So did you use them?
Yeah, so I guess that's a good question. While I was writing the first draft of this manuscript before I shared with the other co-author, I was thinking to apply the repeated measure ANOVA. Yes, that's another option. I can clearly support this idea. Yes, so the repeated measure ANOVA would be really nice to deal with also the correlation, because as far as I remember, the repeated measured based ANOVA also provide the correlation between the time points we are including in the ANOVA. So yes, the repeated measure or random regression, these are the other options can be used for temporal data analysis.
Another question came in, and feel free if you got any more questions. This is our last one. So after he answers this, we'll pause for a little bit while you type. But your histograms of NDVI values per time point look good. Do you standardize the reflectance data besides the reflectance panel calibration?
So yes, standardization is basically happening before we stitch the images. So yes, this is more like a question related to image processing. Yes, we have to do some fundamental steps to optimize the reflected bands. Yes, we are doing a bunch of steps. One of them is the optimizing the reflected bands.
And so the other thing I'll point out here, which has been a subject of some debate so there's some folks here that routinely use traditional measures of remote sensing data, like NDVI, for instance. And in the traditional measures of NDVI, you don't separate the ground from the plant, right? So NDVI, the amount of canopy cover is built into NDVI. So as the plants are small, you get more ground, and as the plants grow and cover the soil and you got canopy closure, the soil goes away, and that's where the dramatic differences in NDVI are. With our group, traditionally what we do is we exclude the soil. So we keep the canopy cover separate from the amount of soil, for instance. And then that allows us to look at the vegetation differences between genotypes more so than the canopy cover, right? And I think either one is sort of exchangeable if you include canopy cover as a function in the model. But that is an important difference between how NDVI is traditionally used versus how we often use it. Another question came in. Forgive the ignorance on drone data, but are the height measurements real height estimates or relative to the neighboring plots? So are they true height or are they just relative height?
These are the heights coming from the 3D point class data from the drone, not like the terminal plan height measurement measured by ruler stick. So if the answer of this question, I may tell.
Yeah, I think that's part of the answer. I think the other thing I would expand on that is height was one of the things a lot of groups do first. And what most people find is, there's ruler measure height, and then there's drone measured height. And those two are highly correlated, but they don't give the same numerical value. And I know in maize, in a crop like maize, we measure plant height by measuring the tip of the tassel and the flag leaf, and we call those plant height. With the drone, nobody knows exactly what it's measuring, but what we think it's probably measuring is the top of the leaf curvature, right? Like the highest part of a large leaf. But when we do the heritability and the genetic variance, when we do the genetic variance estimate and calculate heritability, we find that the drone measured height is better than ruler measured height. Whereas I was just talking to some pulse people a couple days ago, and they actually said that they found that the ruler measured height was more heritable. But they are highly correlated. So they're not just relative to the neighboring plots. They are a measurement of the real height. It's just how do you measure plant height, I think, is the question. And I'm not seeing any other questions, so we'll give people another 30 seconds or so just in case there's any follow-up. Nope. Was that the maximum canopy height of a plot or something else?
We are using the percentile, different percentile, like 90, 95, or 99 per plot. So yeah, this is the, instead of saying a maximum, so our maximum in this case is going to, well, our maximum is 99 percentile derived plant height per plot, but we are using a different percentile. But there are many examples in the literature right now, so the people are just justifying why they're using the different percentile when it comes to plant height extraction. But yeah, we generally use 95 and 99, like how the other researcher has preferred so far, as far as I have seen or read.
Yeah, and just to follow up on that one, for those of you who've never done this, using the drone data by having a stereoscopic image, we can create 3D point clouds of individual points. And so if you imagine in a plot, there's probably, what, Alper, 100,000 to 1 million different points. And so then you just, if you plotted those on a histogram, the very tallest points tend to be anomalies of data processing. So if you took the maximum height, sometimes it'll be, 30 meters in the air, which isn't possible. So the 99th percentile or the 95th percentile, so the top 99, the 99th highest point or the 95th highest point or percentage point we use, because those tend to give heritable, reliable, consistent estimates between plots. This is a very good question, Alper. This is an important one. Are there simpler models to start with to combine genomic and phenomic prediction?
Okay. Well, the answer is so long from my point of view, but since this question is asking for the simpler models to start with to combine genomic and phenomic prediction, so there are really nice examples first. Let me start with that. So the people start with the phenomic prediction by using the NIR reflected lens from the kernel. So, and then they call the phenomic prediction. But the dilemma in this one is, from my biased point of view, is that the NIR is coming from the kernel of the maize, which is what you have already harvested this one, and then you're using NIR to predict yield. So you're already reaching the end at the end of your growth period. So the main point in prediction is how early you can predict your grain yield by using any type of data. Genomic data has been used past 30 years, but when it comes to phenomic data, what we can do is at least we can see some drone feature belonging to earlier time point, and we can use them in prediction. The simpler model, I don't know if this question is asking the application wise or the main idea, but the simpler model is flying drones at the early growth stage, and we can use this data to predict yield, and we can do these things routinely within a prediction within a plant breeding program, like such as public plant breeding program. If you have low in fund, but you can just buy one drone and you can routinely try that. So we are barely trying the phenomic prediction in Texas A&M in the maize breeding program, but not fully applicable right now. But when it comes to application-wise, we are just using the existing method right now. We are using the relationship metrics, we are scaling and centering the data, and then we are creating the phenomic relationship kernel. But the point in here, I would like to highlight that the genomic data is segregating for population of interest in environment A, but the genomic data is same in environment B. But the main point in here in phenomic prediction is phenomic data is segregating across genotype in environment A, also segregating in environment B. These are different between one another, but the genomic data are the same. So this is why phenomic data is a little bit working better as compared to genomic prediction in untested genotype, in untested environment. This is why I showed the last slide. But last conclusion, I have to tell that in the near future, I believe that both technology will disappear. So I'm not defending in here that, or I'm not comparing genomic or phenomic is better than between one another. I'm just saying phenomic data can be complementary. Another thing can be that any phenomic feature can be used as a secondary trait in genomic prediction model. So there are a really nice example in the literature right now. Anybody can easily see them. So multi-trait model, multi-kernel model, these are the application, possible application. We can integrate phenomic data in either as a secondary trait or like a novel kernel, but I prefer to use as a phenomic data as a naive kernel, like genomic kernel.
Yeah, I think that's a good answer. And just to give some perspective to people who may be more familiar with genomics, if you look at the history of genomics, like QTL mapping started with a simple logistic regression. And then we moved into interval mapping and composite interval mapping, doing permutation analysis. Now people are doing Bayesian analysis, right? With GWAS, it started also with sort of simple logistic regression. And then, there was the U and Pressois Q plus K model that came out that was really transformative to eliminating false positives, right? And now GWAS models also are Bayesian and, other AI type approaches. So if you think about those histories and advancements, what I like to think of Alper as doing is pushing the frontier of what, how we can squeeze the most value out of the data we have. But simpler models aren't necessarily bad, but he's really interested in just sort of pushing the boundaries of, what is going to be the future best model. And to me, it seems like there's so few people doing this type of analysis that we're really excited about seeing others, try these models and validate them and develop their own that we can all learn from as a community. So yeah, the simpler model question is very reasonable because you do want to get your feet wet first before his model is very complicated. So you probably want to not just jump right into that. So that's the last question I see. No more coming in. Then I'd like to thank everybody for their attendance and their interest. I know Dr. Adak would be very happy. He helps a lot of people and mentors a lot of people on trying to process this type of data and analyze this type of data. So if you have questions, he's pretty easy to find via email. So I'd encourage you to email him or myself. And thanks for your support of the Plant Phenome Journal and your attendance. Have a great day.